Hello everyone, welcome to Dr. K. PREM-PRIMER Biotech Lectures. In today's lecture, we will talk about subtractive hybridization. See, when tomato plant is grown in normal condition, growth, development and reproduction, that's yield is normal. But if the same plant is encounters abiotic stress, there would be effect on growth, development as well as yield. And after exposing some time, the plant is able to tolerate that particular situation and regain the growth and development and yield will be normal if it is able to overcome the biotic or abiotic stress. So before encountering the stress, after encountering the stress, or during the stress, if you check the genes which are expressed before, during, after, there would be a different set of genes are playing in these three conditions. To know the genes which are playing a role in normal condition or during the stress condition or post stress condition, we need a technique to identify those genes. That technique is nothing but subtractive hybridization. I can give you one more example. You can see raw tomatoes and ripened red tomatoes. If you compare the tomatoes gene profile between the raw and ripened, the genes which are responsible for ripening will be there in the only ripened and not in the raw tomatoes. So but there are set of genes which are commonly there in the ripened one and tomatoes, ripened and raw tomatoes. So by using the subtractive hybridization, we will be knowing what are the genes which are responsible for ripening of the tomato. And I can give you one more example. These are the two conditions. Again, it is a seed germination, different stages of seed germination. Here, radical to the complete germination, the genes which are responsible for each stage are different. And if you can see, if you isolate them from year to year, from radical to the plant, complete germination, there are common genes, there are different genes. So if you remove the common genes, to remove the common genes between the first stage to last stage, if you remove the common genes, and we'll be knowing what are the specific genes from radical to the complete germination stage. That we can do with the subtractive hybridization. In today's lecture, we will discuss about subtractive hybridization, how it has helped scientists to discover the genes which are responsible for various conditions, various developmental stages of cells, tissues, and organisms. So my channel name is Dr. K. PREM-PRIMER. Try to subscribe for more updates. Here we will see what is subtractive hybridization. So subtractive hybridization is a technique The hybridization is a technique that removes the sequences of nucleic acid which are common in the two related samples, leaving behind the different sequences. I told you that raw tomatoes and ripened tomatoes both are same. They are tomatoes, but one is raw, other one is a ripened. If you isolate uh, mRNA from raw tomatoes and ripened tomatoes, and you can see that a set of genes which are responsible for raw tomatoes and a set of genes which are responsible for ripened tomatoes and a set of genes which are responsible for both conditions are there. So when you use raw tomatoes as a, you can remove the common genes which are there in the two stages, and you will have two more sets of genes which are specific to the raw tomatoes and ripened tomatoes. So to identify this differential expression of genes in different conditions, we need to have a technique. That technique is called as subtractive hybridization. So this differentiates between the nucleic acid sequences expressed in two related samples in a specific stage or a condition. When I said in the seed germination stages, radical to the complete germination. Here, they are different stages of the germination, seed germination, right? So you can see a specific set of genes required for radical, a specific set of genes which are required for the completion of the seed germination. Subtractive hybridization mathematically subtracts the gene expression of treated tissues by using gene expression of control tissue. For example, you have a as I as a you as I'm telling you that how do you define the subtractive hybridization mathematically means you have a control condition and treated condition. Whatever genes are expressed in the control conditions, by using those genes or their mRNAs, you can remove the genes responsible for normal condition in a tested or treated condition, and remaining genes are or remaining mRNAs are responsible for treated or tested condition. Remove common cDNAs present in both samples through the hybridization, leaving behind cDNAs unique to the treated tissue. This is what I am saying. Isolate RNA or total RNA or mRNA and convert into cDNA from normal condition and abnormal condition. And by using the cDNA or mRNA from the control condition, you can remove the genes which are responsible for normal function in a tested condition, so that you will have a gene set which is very specific to the tested or treated condition. I think when you will go through the next slide, when I'm going to explain with the examples, you will understand the theme of subtractive hybridization. Here, what is the procedure of subtractive hybridization? You can see the first one is RNA isolation from the test and control condition or control samples. Conversion of control mRNA into cDNA and labeling. Hybridization of control cDNA and test mRNA. Mixture of cDNA and mRNA passing through the HPA column, that is a hydroxyapatite column. Here, subtraction of the common genes which are expressed in two samples happens. So you remove the gene set which is common between the test and the control. That's what happens by hybridization. Elution of the unbound test mRNA and conversion of the eluted test mRNA into cDNA. Amplification and identification of the unique cDNAs. These are the different steps involved in the subtractive hybridization procedure. You can see same thing in the cartoon. I am taking yeast cells are examples here. Yeast cells normally grows at the 28 degrees centigrade. And if you want to grow the yeast cells at 37, what will happen? Grown at 28 degrees centigrade. Other set of yeast cells are grown at the 37. Right? Now you isolate the, the yeast cell grown at the 28 is termed as a control or reference or driver or a subtractor. Yeast cells grown at 37. We want to know how yeast cells grows at 37, and we want to know what are the genes which are expressed to adapt to the 37 or to tolerate the 37. So this is called as a test condition, tester or test. So now we can harvest mRNA from these two sets of yeast cells from grown in at the 28 as well as 37. Isolate mRNA, and in driver condition or control condition, the mRNA is converted to cDNA and labeled. This is the mRNA and converted to the cDNA by using the reverse transcriptase. This is single strand cDNA molecules or which are labeled. Here, on the other hand, yeast cells are grown at the 37. They are test or tester, right? Or target. So isolated mRNA, and you can see the mRNA molecules difference from the yeast cells grown at 28 and grown at the 37, right? Now we will take the cDNA from yeast cell grown at 28 and mRNA harvested from the yeast cell which are grown at the 37. We'll hybridize them. You see, when you hybridize the cDNA of driver with the mRNA of target or tester. Here you take the more mRNA from the tester or target and small quantities of cDNA from the driver. So what will happen? The cDNA molecules which are having the complementary region in the mRNA, they will hybridize. The remaining cDNAs which are not having any complementary sequence in the mRNA, they will remain unbound. Similarly, a set of mRNA molecules which do not have any complementary with the cDNA, they also remain in the single strand. So when you hybridize with the cDNA with the mRNA from the test sample, in that hybridization, there is a population where you see mRNA and cDNA hybrid, as well as single-stranded cDNA, as well as the single-strand mRNA molecules. This is a mixture. Now, this mixture you pass through the, as I told you that this mixture have labeled single-strand DNA, cDNA, and RNA and cDNA hybrid, as well as mRNA. Now you pass through this mixture through the hydroxyapatite column. Hydroxyapatite column, we have a calcium and triphosphates, which will hold the uh, double-stranded DNAs and also have less affinity to the single-strand nucleic acid. Now, when you pass this mixture through the hydroxyapatite column, right? So now you all know that the hydroxyapatite column have a three different nucleic acids. One is a single-strand mRNA molecules, single-strand cDNA molecules, and double-strand or a hybrid of mRNA and cDNA. As I told you that hydroxyapatite column can retain the double-stranded nucleic acid molecules than the single strand. So when you this column is this column is elute with the low strength of phosphate buffer, So with that low strength of phosphate buffer, it can elute these single strand mRNA molecules as well as single strand cDNA molecules. You see, single strand mRNA molecules as well as single strand cDNA molecules. So when you under the double, hybrid of mRNA and cDNA is retained in the hydroxyapatite column. So whatever mRNA is eluted with the low strength of phosphate buffer, that mRNA reflects the specific mRNAs of tester or target condition. Here target condition is what? Yeast cells are grown at the 37. So these mRNA molecules may encode the proteins which are responsible to adapt to the high temperature, and which are these molecules are not present in these mRNA molecules. Corresponding cDNA molecules are not present in the 28 degrees centigrade. Hence these mRNA molecules are not bound with these cDNA molecules. So by using this technique, we can remove the common genes which are expressed at the 28 degrees centigrade and 37 degree, and we can elute the genes which are expressed specifically in the tested condition, right? So here these are the mRNA molecules which are expressed only under 37 degrees centigrade, right? As I told you that labeled cDNA and mRNA hybrid binds to the HPA, which are common between the two conditions of the same cell type. So these are the mRNA molecules. So whatever common are there between the 28 degrees centigrade and 37 are retained in the hydroxyapatite column. So that is subtracted. So by using the controlled cDNA molecule, we have subtracted the common mRNA molecule from the 37 degrees centigrade expressed genes or mRNA molecule. Specific. Now you can see differential gene expression is equal to gene expression in test minus gene expression in control. So here, differential gene expression means genes which are very specific to the higher temperature, that's 37. To know the genes which are very specific to the 37, if you remove the genes which are expressed in control, you use that as a subtract to remove from the test condition. Then whatever is remaining, they are very specific to the that particular condition. So based on the by using the subtractive hybridization, we are using the hybridization technique to remove the genes which are common in the two conditions, and remaining mRNA or cDNA molecules are unbound cDNA molecules as well as unbound mRNA molecules are specific to the that particular condition. You can see whatever common are there are double stranded and they are there in the hydroxyapatite column. Whatever is specific to the 28, they are labeled their cDNA, which are also in the single strand form. And the mRNA molecules, which are very specific to the 37, they are also single strand. Now, these single strand mRNA molecules can be further processed and converted to cDNAs, and you can amplify them and you can clone them. You will be knowing the what are those genes. This is how you can identify the stage or treated condition specific gene expression by using the subtractive hybridization. What are the applications of the subtractive hybridization? Identifying genes expressed at different stages. As I told you that if you take the seed germination as example from radical to the complete germination, and or you say if you say zygote to the fetus, right? Identifying a novel genes. Novel genes means plants which are growing in the extreme conditions, like desert conditions or floods, or in the salty concentration, salt conditions. So if you compare the, it's relevant. It's if you isolate the particular organ or tissue from the particular plant, and you process it, and you will know what are specific genes expressed for that particular condition or particular organ also. Comparing a DNA from different genomes, you can compare the different genomes and you will be knowing the different similarities and dissimilarities between the genomes also, and identifying a genomic region associated with the pathogens. Suppose the tuberculosis have different strains. That different strains are all the tuberculosis strains are not pathogenic. Some of them are pathogenic, some of them are non-pathogenic. When you hybridize both of them, right? So the regions which are hybridized are common between the pathogenic and non-pathogenic, right? But one which are the region which is not able to hybridize with the non-pathogenic one from the pathogenic one means that region may be encoding a pathogenic traits of the that particular pathogen, or pathogenic genes may be there in that particular region. Hence, they are not able to bound with the non-pathogenic regions of the non-virulent strain. So these are the different applications of the uh, subtractive hybridization technique. This is all about the subtractive hybridization. Hope you like this lecture, and thank you for listening. And my channel name is Dr. K. Prem Sagar, or Dr. K. Prem-PRIMER Biotech Lecture. And if you want get regular updates, try to subscribe. And if you have any questions, please let me know through the comment section. Right? Thank you all. See you again. Until then, bye bye.